Okay. Good day, everyone, and depending on from where you are joining us, we are Pharmacy Scholars Initiative, and today is our webinar, which is part of our regular webinar series that we have been conducting for the past 7 years. Apologies, this year has been there's a little bit of inactivity. Most of our key scholars in the PSI academic planning they have so many on their table, and we are unable to deliver most of the activities within the year. But now I think we are all back stronger, and we'll continue with our activities. Apologies for the silence, and we were at the background trying to make ourselves stronger, and I think we are stronger a little bit, and we keep going with where we have started for the past 7 years. So today is 18 October, and I will start with telling you what a PSI is. PSI is a Pharmacy Scholars Initiative, that's the acronym, and is a mentorship network for pharmacists with a keen interest in research and career development. And then this forum was founded in December 2017, and to date, it's registered with the CAC in Nigeria. That means we are fully legal entity, and we have members across different parts of the world. I think we have membership in all the continents in the world. So before we start, we have some announcement to make. This webinar is being recorded as I seek your consent before touching the record button. And then as the webinar is going on, if you have any question to ask, you can use the chat box. And then if you have any issues, you can contact us through our email. And sometimes we have like PSI discussion group. So for those members from there, if you have any question, you can at any point in time post your questions or any remarks. Also, this webinar, if you have something to want to ask at the end of the webinar, you can raise up your hand, and the moderator will point you so that you can make your comment. Also, at the end, we are going to send a certificate of participation to all members or participants who participated in this webinar. A link will be shared on the chat box. So the link is just there to put down your name, and then if you have any feedback you want to give us about the webinar or about our activity, you can drop it on the link, and then your active email address. Then we are going to send you the certificate of participation, and with the speaker's permission, if he permits us, we'll send you the lecture materials. And then from there, you can see our email address there. If you have anything you want to share with us, you can use the email address. And today's webinar, we have some agenda. We have some opening remarks by Dr. Abubakar Shaban, and then the presentation. And in the presentation, we have a brief introduction of the guest speaker, and then we'll give him time to present his presentation. And then we have question and answer session. And then at the end, we present the certificate of appreciation to our presenter. And then we have some closing remarks, and then we'll call it a day. I'll hand over everything to Dr. Abubakar Shaban, who will take us through the moderation. Thank you so much, everyone. Abubakar Shaban. Abubakar Shaban, are you there? Dr. Shaban. Anyway, while we're waiting for
Dr. Jato, I think Dr. Shaban is having some
Okay. I can continue. I'll continue. So our guest speaker today is Professor Mohammed Auwal Ibrahim, and this is a brief about him. This is just a summary. I find it difficult to summarize his achievements. Anyway, He's a professor of biotherapeutics and molecular parasitology at the Department of Biochemistry, Ahmadu Bello University, Zaria. And he's also a deputy director at the Director of Research and Innovation. And he's also the chairman University Ranking Committee of Ahmadu Bello University. His education, he obtained his Bachelor of Biochemistry from Ahmadu Bello University, and I'm glad to inform you he was the best student of the university in the year 2023. He completed his master's from the same university, and he also proceeded to the University of KwaZulu-Natal Westville campus from South Africa, where he completed his PhD in biochemistry. Then he also proceeded to his postdoctoral fellowship in biotherapeutic research group at the University of Pretoria in South Africa. Prof was also awarded a research fellowship by the Japan Society for the Promotion of Science, JSPS, tenable at the Structure-Based Drug Discovery Group, National Institute of Advanced Industrial Science and Technology, Tsukuba, Japan, between 2019 and 2021. And his research interest is mainly focused on molecular biology, biotherapeutics, drug design and discovery, targeting trypanosomiasis, malaria, and type 2 diabetes, where he applied a number of techniques such as recombinant DNA technology, such as gene cloning, protein expression, etc., bioinformatics, cell culture, RT/PCR, NMR, in vitro analysis, and preclinical trials. Professor Ibrahim has authored more than 120 scientific articles published in high-impact journals, and his publication has produced more than 3,250 total citations, with an H index of 34, I 10 index, and of 74 according to Google Scholar. He has also authored 4 book chapters published by international publishers and supervised and graduated 33 master's and 3 PhD candidates. He attended a lot of conferences and workshops across the world. He has also served, where he presented more than 40 papers. He also served as an invited keynote speaker for conferences in Nigeria, Sri Lanka, Thailand, Bangladesh, and Nigeria. In 2023, 24, and 25, he was ranked among the topmost 2% most cited scientists in the world, as analyzed by Elsevier and Stanford University. He has numerous awards. Just to save time, you can see this in your slides when we send it to you. And then he has membership across so many professions. And today, I welcome you, Professor Ibrahim, Mohammed Auwal Ibrahim, who will take us through a webinar on the title Artificial Intelligence in Rational Drug Design and Discovery. Abubakar Shaban, are you back? Anyway, I'll hand everything to Prof. Prof, can you please share your slides so that we can see the webinar immediately?
Okay, thank you, Dr. Zato, for the presentation. You said you don't know how to summarize me. I simply love that is, I think that's an exaggeration. I think the summary is just that that's same hour.
All right, sir. Agreed,
Struggling to survive. Yes. Once again, fellow colleagues, sincerely, let me first start by extending my sincere apology. Let me be sure, can you see my screen?
Yes, sir, we can see it, sir.
On a slide mode, can you see it?
Yes, we can see, sir.
It's now AI irrational drug design. You can see that?
Yes, sir, we can see it.
Okay, that's fine. Thank you. Fellow colleagues, once again, thank you for being here. And let me extend my sincere gratitude to PSI for inviting me. I will say something about PSI in a short while. But before that, sincerely permit me to express my sincere apology for the miscommunication. Largely, I take full responsibility for it. I think what I said to Dr. Jato was that I will be engaged on 18th and 16th. I will be facilitating a program in Kano on 16th and 18th. And I now said that we can have it the following Saturday. Actually, I meant the Saturday after 14th. That is a communication issue. So I took, and also most importantly, I think Dr. Jaton shared the same poster with me. And I only look at my name. I did not take note of the date, and I approve that it will be shared. So I take full responsibility for this. So I'm sorry. It's just now that Dr. Sadiq called me that they are waiting for me. I said, for what? He said this. I said, say, by God's grace, I have some of the slides. I try to synthesize. But the most important thing, really, I would have wanted was to demonstrate how some of the AI tools can be applied. Unfortunately, because of time, I will not be able to do that as much as I will have wanted. You understand? So you have to pardon me for that. But for the rational drug discovery, I think we'll try to say something about it. But the AI component is what I would have wanted personally to expand a lot, to even go as far as demonstrating some data and also some applications. But I think that is what I would say more. I will only, because of time to prepare the slide will take time. So I would simply say a lot of things with that, from that perspective, with just without slides necessarily. Okay, so that is the first thing I want to apologize with us. I will do my best. I'm glad the recording will be there so that people can peer it later. The second point is the PSI. Once again, thank you so much for PSI, and for anybody that is on this platform, please, I will really encourage you to be part of PSI mentorship. I am also a mentee of PSI. I'm a proud mentee of PSI because sometimes this thing that he shared that I was on a postdoc in around 2018 or 2017, thereabout. I was a postdoc at the University of Pretoria. I could remember I came to Nigeria for a break, and I saw a poster in Zaria for PSI organizing something on systematic review. And I quickly called the number that I'm late, but I want to join. And they said I should attend. And I attended the program in Kano, quite insightful, and I'm still in touch with all the organizers. They have been my mentees as far as systematic review is concerned. So I'm also a proud mentee of PSI, a very proud one. And unfortunately, we have not finally written the systematic review, but I can tell you it is through PSI I'm able to pick up so many things as far as systematic review is concerned, especially with we started with Dr. Chateau. He dropped us because maybe I was a slow learner. I was not a slow learner, so you could not move on. But Dr. Ismail did well. We learned a lot, and the only thing is we could not eventually publish any article. And trust me, Dr. Jata, I'll get back to all of you. I don't throw away concept. That folder is still there. The concepts and that systematic review must be completed. So thank you so much. And so I'm just using this to encourage all participants that I'm also a mentor a mentee of PSI. I'm still under the mentorship. I will only graduate when we publish that article. We have started the article. Once we publish it, I will say I've graduated. So thank you. With this, we'll now proceed to what I want to share. It's okay. Okay, yeah. Now, permit me to show you this slide. This is a very important slide. I usually, a lot of time I try to start with this, especially from coming from the background of, I know I'm dealing with so many pharmacy students, in fact, pharmacists rather, both pharmacology, pharmachem, and what have you. And I want to believe most of us are from the Nigerian space. I guess I'm right?
Yes.
Now, there is a very big shift in the area of drug design, drug discovery, and drug development. They are completely three distinct issues in the present day world. It is far much more simpler in a way and precise than the conventional drug discovery approach that we take in Nigeria. Now, the present, which I'm going to demonstrate, the present drug discovery approach far transcends the traditional one that we are currently using, where somebody, I think, largely, if done, simply synthesize a compound or series of compound and start hitting them at anything. That is not very much trendy in the present day world. And will take us to what is rationally used, and that is why it's now called rational drug discovery. So because there is aspect of rationality, and the rationality actually brings about precision, brings about specificity, brings about time, saving time and resources, and so many other benefits. Like I said, the issue of the traditional approach of drug discovery that we are used to, which I think thousands, I would say thousands or hundreds of PhDs are produced from our different faculty from faculty of pharmacy courses. Now we have actually moved slightly above that, and that is what I would really employ everybody on this platform today to realize that we have passed through that, and we should try to also move with the trend. And I've already told us some of the advantages of the rational drug discovery so that we understand. And this is the reason, permit me to say this, this is the reason perhaps why you see a lot of us will produce article will do work, but you see, you find it very difficult to publish in very high impact places because we have been using the traditional method that is being applied in the last 30 years per se, the last 30 years, 40 years. We have moved, and so I want to briefly alert us to where the world is moving, and along the way, I will try to show us how now in the last 5 years, AI is also helping that process. So the story of my lecture will be, I will try to use a model, one of the work I've conducted, and then I will show you some of the limitations, and then you demonstrate to you how AI can come in and also solve some of the problems. Now, I'm sure as pharmacists, you all know this. This is not new at any point to any pharmacy, to pharmacists rather. So largely with this chart, I will restrict myself to the discovery aspect. Before the discovery aspect, there is the design aspect. So I'm going to talk about the design and discovery. Largely, the target discovery, what they call target discovery in this model is the design is actually inbuilt around that area. So this is the general flow, the pipeline for the drug discovery and development. And like I said, we are going to restrict ourselves to the design and discovery wing of it. That is at maximum. I will stop around my discussion. We'll start from beginning to the lead discovery, and perhaps I may touch some aspect of lead optimization as we proceed. And like I said, I would try to show us the practical approach now in rational drug discovery and design. This is my mouse. Okay. Now, this is also a chart that showed that open up the discovery wing first, the discovery aspect of the previous chart. You can see we start in this day and time, we start with target identification. In any serious big pharma, in any serious country, in anywhere recently, the first thing that is usually done is the target identification. And this is what is really done here. What we mean target identification, as I will show us later, is not what conventionally I think we usually do. Somebody would just decide that perhaps he's working on maybe against Alzheimer's, for instance, or maybe against one neuro generative disease, and then somebody would simply pick up just by head decide that this is your target. Now, this is no longer very much terrible. Truly, this is not very terrible. So I will show us a brief of target identification, and then we'll also go to hit identification. Under hit identification, so many things are done, from virtual screenings to real life screening. I will show you. I've exported the slide. It's a demonstration of some of the screenings we conducted, so that we see how rationally you arrive at a particular group of compounds. And then, like I said, I would integrate AI along the way as we proceed. Then hit to lead. After identifying hit, you now identify lead. This is where de novo design comes in, around here. Sorry. Yeah, this is where the design comes, because from identifying hit, you now have to design the lead now. Now you now further optimize the lead. I will show you how systematically we are able to achieve this in the next few slides. So I would say that based on a number of experiences and based on a number of I say examinations I've been to, I think we are yet to, as pharmacists, we are yet to start following this approach. We only, I would say, I know, so that you don't crucify me, I know we always do it in classes. I know we always do it in classes, but when we come to research, I used to find it hard that we actually don't pursue it as much. I'm aware also of our limitations from the sources perspective. Yes, but I think some minimal effort, if put, can actually help a lot. So that is that. So based on this, I will try to look at generally two aspects of rational drug design. We have what we call the structure-based drug design approach, and we also have the ligand-based drug design approach. In structure-based drug design approach, this is where the structural data of the target is available. I will show us also in a few slides. Now, in SBDD, you have the target, the structure of the target is known. So if you know the structure of the target, the next thing is for you to go and find out, is for you to get the target and also conduct your studies with the target. Like I said, that simplifies so many things. In fact, with structure-based drug discovery, a lot of side effects that we see later are actually minimized. We can't say for sure that it's zero, but they are absolutely minimized when you use rational drug discovery approach. You minimize downstream during clinical trials and downstream applications. You minimize the chances of side effects. And also we have the ligand-based drug design, where the structural information of the target is missing, but you can use information about the ligand to start designing your drug. So those are the two major design systems that are used. And of course, in the case of SBDD, we rely on knowledge of the target structure, and that target structure you actually have to use especially X-ray crystallography to obtain the NMR. Of course, usually not the NMR for elucidating compounds. I will show us an example perhaps. You use also NMR to also not NMR necessary, any biophysical technique from NMR to SPR, that is surface plasmon resonance, and other also protein thermal shift assay. Those are biophysical techniques that are used generally in understanding in understanding binding, the process of the interaction and binding between the target and also the corresponding ligands. So that is the basics. Generally, I would say I would prefer to look at drug discovery, drug design and discovery from these two perspectives. Now, with this very basic background, I will want to now tell us that I've been talking about target identification. Before I start demonstrating some examples, I've been talking about target identification. Now in with advent of AI, now machine learning and deep learning techniques can actually be used to predict very suitable target. Like I said, some of these AI issues, I'm going to just show one slide and then I'll give as much examples as possible because I will have wanted to zoom in here. Now here, you can see you can use machine learning, deep learning approaches, and you analyze complex genomic and transcriptomic and proteomic data to identify disease-relevant targets. Here, what we are saying is that AI and machine learning has the power that you collect very big information, complicated information. Let me give an example. For instance, there is an outbreak, for instance, maybe an outbreak of a disease. Now, you can collect complex genomic and transcriptomic and proteomic data and subject it to machine learning. From the machine learning algorithm, you will be able to depict some unique features of the disease, you understand, that have not hitherto been identified. Machine learning does very well in this, especially coming from what we call multi-omics approaches. Machine learning does well. Now you're able to predict very new features of a disease. Now those features, that tells you that, okay, if this is something that is quite unique, it means therefore I can now, and of course, after validation, of course, you can be able to predict that, okay, now this unique feature of the disease can allow me start exploring that as an avenue for drug development or for drug design and discovery. Let's say, for instance, like I said, let's say there is an outbreak of a viral disease. Let's go with viral disease. The viral disease, then you are able to get the virus, analyze, conduct extensive genomic analysis of the virus, conduct extensive transcriptomic analysis and also proteomic analysis of the virus that is in circulation. Then you subject that data to machine learning. Usually machine learning has ability to pick out things that are quite unique. You pick something that is absolutely unique to that virus that is circulating, let's say, in northern Nigeria. Once you are able to do that thing that is unique, you will now go further to consider, that is consider making some other considerations to now see if that can serve as a very good target. And then subsequently, as soon as we are able to identify maybe perhaps a particular protein is a very good drug target, then the next thing is to move to the next step. Get your protein and test, and then from there, if things go well, you end up with a drug specifically targeting that strain of the virus in question. That strain of the virus. So you see, with AI, with machine learning and deep learning techniques, rather than spending months using conventional bioinformatics approaches, machine learning can simplify and identify target far much more faster than conventional bioinformatics. And it is also contextualized. What I mean by contextualizing is because you will be able to identify target that is precisely relevant to a particular, even not species, to a particular strain. Let's say, for instance, maybe I don't want to say COVID-19, but we can use it because I know viral diseases are more emerging. They are more emerging, already emerging. That's why I prefer to give example. Let's say even, for instance, Ebola virus. There is, God forbid, we have an outbreak. Now we can extensively study the multi-omic profile of that virus, and with that we'll be able to see the difference of we'll be able to identify new target that is hitherto not identified in previous Ebola outbreaks. And with that, we'll be able to design precisely, and I mean precisely, a drug that will target this strain of the virus. So with this, and of course, also maybe our questions and comments will come up so that I can expatiate on some of this point. And of course, perhaps drug, we can apply it because of mutation that the virus might have undergone. We can apply some drugs that are known to address Ebola virus elsewhere, may not necessarily be applicable in a particular strain. Of course, I'm talking to experts, so I'm sure you understand some of this mutational problem that lead to drug resistance and what have you. So AI can actually help you to achieve that far much more faster than conventional approaches. That is especially machine learning and deep learning approaches. Just get the data, subject it, it picks up very novel targets for you. Just tell it tell the model to identify very novel targets for you, and then from there you have your unique target for a strain, not even a species, and then you go further downstream to start considering drug design, designing drug. Now with this, it means therefore AI is crucial now in facilitating, in simplifying, in speeding up the process of target identification from this perspective. I know I will repeat this slide later in due course. Now, I want to take us now to what we have been doing. Like I said, I will be using this model to be integrating how AI becomes paramount, how AI in the last 5 years has revolutionized so many approaches. Now, fragment-based discovery is also, some would say fragment-based drug design, is also a form of structure-based drug discovery technique where we try to construct, where we use fragment to construct a lead compound. And like I'm sure colleagues will remember the first chart I've shown us, where I show us hit to lead, hit identification and hit to lead. Now, fragment-based design and discovery approach will allow you also systematically identify target, and then from there you use fragments of compound to have a lead compound that will completely knock off a particular protein, and subsequently you design you move further with your drug design process. Now, so of course, drug design is partly why it evolved is because of combinatorial chemistry and also high throughput screening methodologies that are used in so many pharmaceutical industries these days. So now with this, it provides a more efficient system to screen collection of small molecules. That is what we call fragments, and subsequently you expand them, or you match them, or you link them. That is the lead designing the lead. Now from there you can now move further. I think I will summarize this. This is largely what I just said. In the fragment-based drug approach, generally there are 3 distinct stages. I think I'm going to take us through each of them. Now, first, in fragment-based drug discovery, there is, apart from target identification, which I'll also come back to later. Now, you also have to generate a library, and once you generate a library, you validate your library. That is the second stage. Then, after validating, or rather, you screen the library, and after screening the library, the next thing is you now use the information that is derivable to design your compound that is quite unique to that particular context, or that particular protein, and also that particular disease. I'm trying to save time. Now we move to the first step of the fragment-based drug discovery. That is what you call the fragment generation. Here we apply what you call the rule of three. I'm sure colleagues are well aware of the Lipinski rule. That is actually a rule of five. The rule of five of Lipinski no longer very much applied to so many drugs that are coming up into the market. And that is some of the information I want to bring forward to my colleagues in the platform, that the conventional Lipinski rule of five do not apply as much these days. In fragment-based drug discovery, we use what you call the rule of three. We all know, rather, I don't know that we all know, sorry about that, but I guess most pharmacists know the Lipinski rule of five. I think the rules are there. They are not very much applicable in rational drug discovery now. Rather, we use the rule of three. I will show us why later. So in fragment libraries, generally we select fragments. That is why at this stage we don't call them compounds. We call them fragments because they generally obey the rule of three. Now, okay, I'm just saying that this is not applicable. Now, in the rule of three, the chemical compounds are quite available. The chemical compounds that obey the rule of three, they are quite available in so many platforms. We have commercial ones, we have those that belong to some pharmaceutical industries, those that are, you can see those that are close. For instance, Zinc database is one very interesting database that you can recover fragments that are that obey the rule of three. Also, Chebi is also another platform. Also, apart from that, some pharmaceutical industries have also created their own libraries. For instance, the Maybridge rule of three fragment library, the Kishida library, the Novartis library, they all have generated their rule of three library, because the reason why I think this is, especially from the pharmaceutical industry's perspective, is important here, so that some of us will realize that even the big pharmas, actually that's what they use now. I don't know eventually what they brand, but eventually from the starting point, usually they have their fragments that they generate. Usually in some cases, you see the library can be up to 30,000 fragments. Some I would talk about 100,000 fragments and so on and so forth. Now, after the fragment generation, you also do fragment heat identification, and that's what I think I showed us in the first earlier chat. In heat identification, we use direct functional or direct binding assays to screen, or you can use NMR binding assays, or we can use crystallography-based assays. I'm going to give us some specific examples as I proceed in the next lesson. But the most important here is this is where you are able to identify a particular heat from your fragment. And after that, you link your fragments depending on the nature. I'm going to show us an example. Depending on the nature of the compounds, you link them based on certain features, you link them and you have a hybrid molecule. Now, this is where the de novo design is, where you will produce a lead compound that very precisely, and I mean very precisely, inhibit your protein and inhibit your protein only and only your protein, because you design it de novo. You cosmetically design it, if I can use that word. You use cosmetics to design it, and as such, it is quite very precise. And now these are some of the successes that have been recovered. Some of the successes of fragment-based drug discovery. Some drugs are already in the market, like Zelboraf. It's very difficult. I find it. Zelboraf is actually anti-cancer drug. It's actually generated and produced through this process. And also Onales PIP is also another one. Also the venetoclax. These are all, I'm sure, as pharmacists, I'm sure this is not new. I just want to inform us that this these drugs that are quite very useful these days are not produced through the conventional and traditional drug discovery that we are used to. They are produced through, this one specifically are produced through the fragment-based drug discovery. You can see the moieties, you can see this very interesting moieties. They are actually constructed directly, not that it's not just by accident. They are constructed from day one. Now, at this point, this is why having provided that background, it is at this point I want to now shift to, like I said, what we have done from rational drug discovery perspective. And along the way, I will show us how AI is helping to revolutionize some of these processes. Now, specifically speaking, the first thing we did was to identify a protein, a very important protein that we think is very relevant to the survival of a trypanosome. This is GPI phospholipase C. It's a very important protein that tries that plays very important role in antigenic variation in trypanosome. Of course, I believe you understand antigenic variation is a process whereby the organism keeps changing its surface coat as it's being exposed to immune pressure in the host environment. So in order for the organism to keep surviving, it keeps switching its variable surface glycoprotein, and as by so doing along the way, it keeps changing like the coat, and as it changes the coat, it allows it to keep evading the host immune system, and by so doing, it continues to perpetuate the pathogenic process. So we became interested in that very important protein because it is the key protein that allows for switching of the switching of the parasite, and of course enhances its pathogenic processes. So this is the first thing that is usually done, that is largely neglected by our traditional drug discovery approach. Once you now identify that you want to work on a particular protein, the first thing rationally to do, and this is, like I said, what is done in most of the big pharmas, you try to either get the synthetic gene or whatever technique that you can use to identify the recombinant protein. Identify the recombinant protein, and perhaps that is why whenever I said that my field is drug design, and people will see that I usually start with core molecular biology with recombinant DNA technology, I know pharmacists will always keep, I said, do I say crucify me? You understand, in Nigeria, that why this is not pharmacy. You understand? I think I received this a lot. That this seems not to be pharmacy, because this is usually the starting point in most big pharmas, like I said. Now here, I will summarize. This is all about recombinant DNA technology, where you get the gene. Here we get the gene. In this situation, we now try to clone the gene, we express the gene, and eventually we transform an E. coli to express the particular protein of interest. In this case, we are interested in TBPLC, trypanosoma brucei phospholipase C. Now, this same or can actually be transcriptomic and proteomic information that I've discussed earlier, where the process of the identification. Now, after we identify that, okay, we are interested in TBPLC, we now we now clone and we express the protein this way. Now, after expressing the protein, this is just to show us that the protein has been successfully expressed quite successfully. We can see the protein, and so we are happy. We can now move further to play around with the protein. Now, I'm bringing this cell chart back to show us that this process that I've shown us, usually, personally, I think depending on how fast you are, that can take you up to 6 months. You understand, with lots of resources, starting from the gene, moving to expressing the protein, leading to purifying the protein. I've even jumped that part, purifying the protein. That can take months, depending on the nature of the protein. 6 months, 3 months, and if you are not lucky, it can take you up to 2 years. In standard places, even up to 2 years to achieve this. But and that's what will allow you to have your particular protein. Now, but with AI now, you don't necessarily have to go through that complex process. You can, after simply from the genomic information of the virus, you can simply look at what is quite unique, and so as you now see what is quite unique, you can just proceed and work with it. So the complex process of cloning, transforming, expressing can be, to a large extent, or rather is largely simplified with the advent of AI. So that is one thing that is being applied. Like I said, I would have wanted to expatiate on this aspect. Now, like I said, this is what I want to be showing us, how difficult it was and how simple you can integrate AI. So this is one way, rather than to go through the complex process as I went through, you can actually use AI to simplify the process. Now, after we have our protein, as I've shown us in the earlier slide, we had our protein, and this is where the issue of fragment, as I've shown us, comes in. We generated our library from the Maybriggs rule of 3 diversity fragment library, and also from another library from Kishida. We generated about 874 fragment compounds. We call them fragment compounds because they are not compounds under the situation now. They are not compounds. So we subjected those 8 fragments into conventional biochemical analysis to see how much they are able to inhibit, to get into inhibit the catalytic activity of the protein. Now, having done that, we subjected 874 of them into a high throughput screening assay. You can see eventually only how much? Only the red ones, they are 30 something, actually passed through our set criteria. And therefore, after that, you see those are the red ones, and then those 66 rather. I don't know, I've animated it, sorry. There's those 66 primary hits, you call them primary hit fragments. You are selected. Now, after selected them, this is where we now take them to what an experiment called waterlogged. Now here, it must not necessarily be waterlogged assay or waterlogged NMR assay. It can be any biophysical assay, ranging from surface plasmon resonance to protein thermal shift assay and other NMR-based systems that will allow you measure the direct binding of the hits to the recombinant protein. Now, and that is just this chart to show us what is waterlogged. So all the 66 primary hits that I've shown us earlier, actually we subjected them to this waterlogged NMR to see how are they able to directly bind to the hit fragments. Now, on doing that, we recover that this is the system, the waterlogged result of the 66 fragment hits, and among them, only 10 actually bind to the protein. It means, therefore, all the other ones are false positive. Now, this is also something very important as far as rational drug design is concerned. You see, in our setting, what we do is that we just hit let's say, for instance, you are working on antihypertensive candidates. For instance, you are targeting angiotensin converting enzymes, for instance. Now, if you just take your synthetic compounds and and see, test it, and that's what is commonly done, of course, and test it. Usually, a lot of the results that we actually report, I'm sorry to tell us, are actually false positive results. Most, okay, do I say most? Or conservatively, let me say some are actually false positive results. This is what I show us is a clear demonstration. We use conventionally what we are used to. You can see among all of them we had 66, but on subjecting them to analysis, to about physical analysis, we are able to only understand that only 10 actually can serve as drug, can actually bind, can actually bind to the protein. You understand? All others, what they produce is actually false positive. And this is some of the limitations of the traditional drug discovery that we are used to. Without it excludes us from having this precision. You can see this is quite very precise. We are now almost unequivocally sure that these hits are actually what we call true hits, and they actually bind to the protein. So with this, if we move further, we have demonstrated unequivocally there is no need of going back, and we don't expect any false positive result down the line as we proceed with our drug discovery studies. This is the compounds. You can see them, not the compounds, permit me, sorry. These are the fragments. You can see fragments, it's 1, 2, 10 of them. These are them. They all obey the rule of 3. They don't obey the rule of 5 in the case of Lipinski. These ones obey the rule of 3. Now with this, we now see the compounds, and we now state, okay, let's now go back to conventional bioinformatics to see how do these compounds actually interface or actually bind to the protein, because we have now seen from the NMR that these are the only ones that actually bind to the intestine, to the protein. So we now subjected them to further bioinformatic work to see how they bind, and eventually we now realize that just trying to summarize because of time, we now realize, you can see this very important information, that fragments 6, 7, and 9, do you see this is where they bind? I don't know that I can draw. Okay, I cannot draw. You can see fragments 6, 7, and 9, you can see where they bind. You can see fragments 1 and 8 where it binds, and also fragment 4 where it binds. By this message, it is now telling us, now this is the fragment-based design. This is now where we will now design a lead compound unequivocally. We know the active site. We have seen that those from the waterlogged NMR assay, we have seen that they bind. Here we see how they bind. And with how they bind, this is clearly telling us that if we want to have an effective inhibitor of this protein, we should match fragment 6, 7, and 9 with 4 and 8. So you can see we can have so many leads. For instance, if you combine fragments 6, 4, and 8, it will lock up the active site because you can see how they interact. Or you link up fragment 7, 4, and 1, or 7, 4, and 8, you also have a lead. Or you now pick 9, 9, 4, and 8, or 9, 4, and 1. Now, these are all potential leads now. So you can see how we use fragments to have leads. Now with this, we are now having lead compounds. Sorry, from our fragment hits, we can design a lead, and from there we move forward. You can see from how you can construct, and this is what we are talking about: de novo drug design, or de novo design. You can see we are using fragments to design a hybrid that is large enough and also rationally designed to fit into the compound. And with this truly the next thing is just to move further. It's just to move forward. You have rationally now design a lead compound. That is the rational drug design that we are referring to. This is, I'm sure colleagues will believe with me that this is quite a bit different from what we have been doing traditionally, where it's when we design, sometimes you go and start looking how it block, but it is rationally, it should be the other way around. Now, I think this is the summary of all that I've said. We have published some of this information, and I think I will summarize. These are some of the challenges that I will say AI has solved now. Now, we try to crystallize out the protein, because remember I informed us that after you have the protein, it is important you also crystallize part of SBDD. We try to crystallize it, of course, to a certain extent, I would say not successfully. I'm saving time. And also we subjected it to cryo-EM. We subjected the protein to cryo-EM, also tried to elucidate the full structure of the protein. It was not working very much. And this requires complicated techniques. I think cryo-EM system, one can know how many cryo-EM systems are available in the entire world. Yeah. And also even the X-ray crystallography, the diffraction, one can pick. For instance, I think let me talk about where I know. For instance, I know in Japan there are only two centers where you can successfully conduct X-ray crystallographic analysis. Also the same thing with cryo-EM. Now, these are very complicated, requires time, resources, and so many issues. So many issues and so many challenges here and there. Like I said, I think I've spent nearly 2 years on this project, and I will tell you for free that I've not finally successfully even crystallized it out, even though I have some very fantastic images, but it is not final as we'll have watched. That is why we switched to cryo-EM to see what we can obtain, but also without much success. Now, this is now critical information that defines the recent approach to drug design and discovery. Rational drug design and discovery, like I said, a bit different from our traditional approach. Now, with all the complications of this system, where does AI comes in? That is the next thing I want to take us to. Now, with AI now, unlike also conventional bioinformatics, AI will help you simplify all these complex processes I took us through using deep learning models. And I would specifically talk a lot about AlphaFold, which was developed by DeepMind. And I'm sure a lot of us might have read a lot about the AlphaFold system. Now, about all this thing that I've talked to you, spending 3 months, not nearly 2 years, albeit not very successfully. Now, AI tools have come up that have simplified this process, that you can get results within a day to move further. Even though, of course, especially to crystallographers will tell you that it is not 100% as expected, but at least it is far better than the conventional bioinformatics, than the conventional approach. It simplifies the process by, like I said, maybe from 2 years to a single day. That is quite huge, and it's AI that does this. This is a predictive model for the target protein. So by this, what AI does, especially the AlphaFold, is that once you have your protein, once you have identified this is the nature of the protein that you are interested, this is, in my case, let's say for instance, is TBPLC you are interested in, you can actually model the structure of the TBPLC with AlphaFold. This is quite, you can see, after, rather than spending 2 years, you can actually do this with AlphaFold within hour, within a day, you can now predict the structure, but we said it's prediction. So it's still not as versatile as confirmatory as the other model that we have used. Now, so this is one very critical point that the AI is assisting us in rational drug design and discovery. Like I've said, that is why in my topic I did not say in drug design and discovery, because so that we don't expect the traditional approaches. You understand? In this rational drug design and discovery, these points have shown us the other one in target identification, where you can use genomic data and machine learning to for AI to facilitate the process of target identification. Here also AI can help you in predicting the structure of your target, and that makes things very fast. Also, I've shown us, let me take us to the other slide. Just take us back. I've shown us how we took a lot of time to arrive at this figure. You understand? Because it's this figure that gives us hints as to which direction to follow to design our lead rationally. So with also AI techniques these days, you can use generative models and what they call also reinforcement learning to design novel drug-like molecules with the desired properties from scratch. You see, this is exactly what this is. This chart, that is what we did. We designed drug-like molecule from scratch. Now, this is what AI also can contribute to using generative models and reinforcement learning. Like I said, the AI component is a bit weak in my presentation because of the communication issue that we had earlier. I did not, but I can speak to it a bit. But the slides are not are a bit scattered. So you can use these generative models to now, in relation to your AlphaFold, to input your data. And after you input your data, the AI will now process until it will produce a drug-like molecule for you. And then from there, of course, you can also further subject it to further optimization process. This is the lead optimization stage, where you look at efficacy, solubility, and other admit properties. So these three fundamental stages are where you actually can apply AI in rational drug design and discovery. From target identification, I've shown us what we do and how AI can help. And from the construction, I've shown us where, that is the other slide. Sorry, in the prediction, I've shown us how challenging it is traditionally and practically, and where we are, how AI can facilitate that, and also in the construction, where AI can also assist us and facilitate the process. And I think with this, I would say thank you. I wanted to ask about how much do I have, and I think thank you so much. I think I need to summarize at this point. Thank you so much, and I can take questions, comments, and observations. Thank you.
Thank you so much, sir, for this brilliant presentation. Though it's caught you within, I would say you are not really prepared for it, but just because of your love for PSI and you also love to disseminate knowledge whereby you are called on, we truly appreciate, sir. And I'll hand over to Dr. Mustafa. Mustafa, are you around so that you can take over?
Yeah, so thank you
And
so
then
much.
before Mustafa will continue, yeah, sir, I have my own questions, but I'll allow the moderator to to take over. And then the only comment I want to make is, yes, sir, we really appreciate you were there physically during our inauguration from the first day we started in, I think, December at BUK. I physically interacted with you, and we really appreciate, sir. And sir, you are not a mentee, you are a mentor. Thank you so much, sir. And Mustafa,
Thank
please.
you.
Okay, thank you so much, Professor Auwal, for this very insightful presentation. I want to believe for most of us, we have been taken like 5, 10 years back when we are in School of Pharmacy studying some of these things, medicinal chemistry, pharmaceutical medicinal chemistry, and it's quite refreshing. And I'm glad to see a lot of advancement in this field. And thank you so much, Prof, for this knowledge. So we are going to take some questions from the audience. If you have any question, you can indicate by raising up your hand to ask your questions directly to our speaker, or you can as well drop in the chat box. I can, on behalf of you, read out the question to our speaker. I think I've seen one question from
Yes, you have one
Muhammad.
question there.
Yeah, from Muhammad Yalwa. So, yeah, I've seen Dr. Jato raising hand, but before Jato, let me just read out this. Then after this, Jato, you can ask your own question. So he said, thank you, Prof, for the presentation. His question is, after you successfully discovered your drug using AI and DML, is there the need for preclinical trials or not? And what is the limitation of AI and DML in drug discovery? This is from Muhammad Yelwa. So, Prof, if
Okay.
you can respond
Yes.
to it. Yeah, thank you.
Yes. Thank you, Mohammed, for that question. Now, let me tell you that yes, you actually will need sometimes, depending. Sometimes there are cases where people from here move to clinical trials, but I think it's a bit risky. I think a lot of us also prefer testing at the preclinical stage. But one fundamentals that you should know is that at any rate, whether you subject it to preclinical trials or not, the using AI as I've demonstrated actually reduce a lot of side effects that are known to be associated with that usually come up when you use traditional drug discovery system. That allows you to drastically minimize side effects or even failure. For instance, in fact, okay, I should say that first. For instance, you increase the specificity and the potential potential of the candidate to become a drug when you use this model. This is far much more superior than using, like I said, the conventional approach, and that is why you see even those days, not those days, even now, we say that when you start maybe with 100 compounds, before you reach clinical trial, maybe you have 2 or 3. I think that's right? And then maybe even after the clinical trial, you end up with 0. Then you go back again. Now, but with this approach, you drastically minimize that chances. That chart, I think I have not shown us that chart where you see the process of drug design and discovery start with so many hundreds, and then you end up with 1. Now, with this approach, you drastically reduce some of those problems. You drastically reduce them. You see that maybe you start with 5, eventually we end up with 1. You see that has reduced time, resources, and so many other things. So that is the issue of clinical is actually, I would say, is a lot of times is truly needed. AI is still not yet there to completely replace some of this technique, but it facilitates, it makes them faster and easier. That's what it does. It does not completely replace so many things. It's just like saying it does not, AI so far has not replaced human brain. That is exactly the analogy. That is where the analogy to it. And then what are the limitations of AI and DML in drug discovery? I think the limitation is that it is still predictive, even though it predicts with very high degree of precision. But I think the main limitation is that it is still predictive. There are some situations whereby the result may not necessarily be correct, or it fails in some situations because it is still predictive. But what we are saying is that it is predictive, but with very high degree of precision, and it increases specificity and it reduces the time and resources consumption. I think that is that. I hope that answers it. And of course, I think I must say that is one thing I wanted to add. Of course, if you go through this route, the chances of you succeeding in clinical trial also becomes very high, rather than you take the conventional approach. Thank you.
Yeah, thank you so much, Prof. Auwal. I think that's very clear, and I'm very sure you've already addressed the question. So I would like to also call upon Dr. Ibrahim Jato.
No, I think finish the ones on the chat box.
Sorry?
I can finish the ones on the chat box.
Okay. Yeah. So
just a comment from Mom.
There is one question, right? Are there special AI courses that a pharmacist need to take before they could apply ML and deep learning in drug discovery and design?
Specifically, I think a number of courses do exist, and some are what do they call them? Subscribed, you understand? But sincerely, I to go over them. I know I've registered for some of them, not long ago, even though I've not fully committed myself. But I think in a nutshell, I can tell you that actually they are quite available. I think you can once you simply Google, you can be able to recover some of them. So they actually exist. And one thing I want to also tell us, which I also neglected, my talk, I always try to restrict myself to the design and discovery. I try not to jump to development side. Dr. Jato, I know, is mostly in the development phase during looking at clinical trials. So I try to always run away from that. That is not within my domain. But one thing I can tell you, AI also plays very important role in the development phase. That is from clinical trial upward. It simplifies so many other processes. And I think a lot of the AI-based courses are more inclined to the development stage rather than the discovery stage. Thank you.
All right. Thank you so much, Prof. There is another question from Abdullah Ahmad. That does the compound obtained through this new procedure undergo ADME tox prediction?
Yes, it does. But again, I will tell you that usually you don't. Once you follow this, the chances of them failing the ADME prediction is very minimal, if not barely non-existent. Very minimal. You minimize that. But I think for clarity, you still subject them for clarity. But one thing is that you are very less likely to see that they fail at that stage. But I think you still push it just to be sure. Like we said, AI and this procedure is said to replace humans on some of the approaches, some of the scientifically tested approaches.
Yeah.
Thank you.
Thank you, Prof. Ahmad. There is another question, but I feel like you've already addressed. But unless
yes, I think so.
Want to chip in,
so I think I've emphasized on that one.
Yeah, regarding the
efficiency. The only thing I would say limitation of my presentation, which is because of the challenge, is that I've not given some very specific sites and also, some specific sites. And also, my initial plan was to even do some 5 minutes, 10 minutes demonstration, honestly. And I think that is one major limitation of my presentation. But I think I've provided clear evidence on that this actually improve efficiency and accuracy. Yes, thank you.
Yeah, so I also agree with Prof. Auwal. Maybe during the course of sharing materials, if you can add some of these links, especially to the
Yes.
course, to the another person asking about library, I'm very sure maybe when we're sharing the materials, we can have links to the fragments libraries that they can
Yeah,
always.
for the libraries, I think I've listed some in my slide already. Zinc database, Kishida, Novartis also. But for the pharmaceutical industries, Dr. Jata will understand that for pharmaceutical industry, a lot of time they close it off. It's not public, it may not necessarily be publicly available. For Novartis, even for the male breach rule of three that I've used, I will tell you, it's under contract that we obtain it and with strict regulations that we obtain and use it because it's from a standard big pharma. But there are others that are available on the publicly available database. Like I've given us example of Zinc database, of the CHAPI, of DrugMark, and others. Thank you.
Yeah, thank you, Prof. There is this question also, maybe I'm very sure maybe a lot of people also want to ask. Someone is asking regarding the conventional AI tools. I'm sure they are talking about ChatGPT, Gemini. So can they also give hint on the toxicokinetics profile of the leads?
I would say to a very limited extent. You understand? They, of course, as you can rightly predict, they will not be as robust as models that are specifically designed and trained for this purpose. You understand? They cannot be as robust as those ones. But what did I do? I think it was in Kaduna. I tried to do some things actually with AdMed, and I think the results not 100% distant, but it gives an idea. So you can actually do very basic, but don't rely so much on it. It's not as robust as when some models that have been designed and trained specifically, designed, trained, and also validated for these processes. It is not as valid as that. Recently, I would say, I don't know whether you hear, one of my students, that's what we are trying to do. Specifically, we are trying to design an AI model for predicting GDM, for predicting type 2 diabetes from GDM in northern Nigeria, so that if we have an AI system that we know certain percentage of GDM mothers, gestational diabetes, that's what I mean by GDM. We know certain percentage will have tendencies of progression to type 2 diabetes later in life. So what we are doing is to now try to develop an AI tool that will allow us, once we are able to know that a woman has already developed GDM at any point in during actual, what do you call it, that stage during delivery. Sorry, when they have the ability to conceive. Now we'll be able to now model to know that, okay, when you reach 60 years, certainly you come up with type 2 diabetes. Or when you reach 55, you come up with type 2 diabetes. So we start to prepare people. That is, so you see, perhaps you may be able to do some of these things with the basic ones, but will never be as robust as the one we have already. We get data from within, we develop the tool, we validate, we train it, we validate it. It cannot be, nothing will come as close as to the AI model that we'll develop. But the general ones still remain general, just for idea's sake, but not as robust as this ones that, for instance, we are able to develop. I hope this clarifies this thing, sir.
Yeah, I think that it's very clear. Thank you so much, Prof. Auwal. So maybe for the purpose of time, because we are already very, very late, we'll just take two questions from the chat, and then Dr. Jato, from your question, we can round up. So just quickly, one question from Dr. M.A. Hassan. What are the ethical issues and biases with the use of AI in drug design, especially with with complete models that are difficult to interpret. And then he asks about also, do regulatory bodies provide guidelines for the use of AI in drug design process?
Uh, when I read this question, I laughed. Dr. Jato first sent me message that whether I can give talk on ethical issues and ethical use of AI in research, something like that. I told him that it's a thorny issue. I will run from it. I will run for my life from giving that talk because ethical issues and AI are still quite thorny. You understand? So I try as much as possible to run away from such controversial discussions. That is why I told him, that's why I said I'm not going to give that talk. You see, Sim God will have it. It's also coming back here for me to say something about, and I think I will still stick to what I said. It's quite thorny. I'm sure we all have agreed with the fact that there is no globally accepted framework that defines ethical issues in the use of AI. I use globally accepted framework. I know there are other frameworks that are in bits. Dr. Jato, perhaps I'm right.
Yes, sir. Yes, sir.
Some of the ethical frameworks are actually in bits. There is no globally accepted one yet. And that is why I prefer to keep mute because some of these fragmented ones come with elements of biases because some of them, I say about some of this in publicly. Of course, some of them may be pushed with some elements of conflict of interest. You understand? Conflict of interest here and there. And that is why discussing about ethics, I think, in my opinion, is still thorny, it's still a bit controversial. It's still a bit controversial. So I would say that I would still prefer not to say so much as far as ethical issues are concerned with AI, especially talking in public. Yes. I'm sorry I did not answer this well. I know.
No, I think you are very clear, Prof. Yeah.
I want to be very careful, yes.
The area is evolving, so we believe with time it will be clearer. Yeah. So thank you so much. Then the last question, sir, in the case of ligand-based drug design, where the structural information of the target is missing, how do you go with that?
Yeah. If the structural information is missing, that is where QSAR comes into play now. QSAR, that is where it comes into play. I'm sure most of us know QSAR. I try to run away from that, maybe because of time, and also this is what I can quickly gather within the 10 minutes I requested. QSAR is the quantitative structure. QSAR is actually what is used in designing ligands where structural information is lacking. How it works is simple. By collecting information about ligands that act on a particular drug, in some cases, sorry, I said ligand or working on drug. No, I mean ligands that, or rather drugs that are not to target that classes of diseases. Let's say, for instance, you are looking up, you are thinking about diabetic candidates, and you are interested in developing using ligand-based system. What they do in QSAR is first to collect all the drugs, all the chemical scaffolds, rather, all the scaffolds that are known to act on the disease. Then they start modeling with QSAR. And then with that, you arrive at putting so many things into consideration. That's what QSAR do. Putting so many of them into consideration. Eventually, you now can come up with a lead that is fairly versatile, that takes so many things into consideration. that is, of course, you can see that without the structural information, you are able to use legals to have something. But of course, that also has to go with testing. Now with AI also, AI can also facilitate the process of the QSAR process. Like I said, I did not discuss much about this, but the process, the QSAR technique can also be facilitated by AI as we proceed. So that is, I think that is the response.
Yeah, so thank you so much, Prof. Auwal. I think we'll just take the last question from Dr. Jato, and then the next will present a certificate, and then we'll just round up. So Dr. Jato, if you are still
Okay,
ready.
yes, I'm here. Thank you so much, sir, for this excellent presentation. We really appreciate it. We learn a lot. I have two questions. My first question has already been addressed. And the second question is partly away from the main topic today. Sir, in the beginning of your presentation, you mentioned something that sounds big to me, like the traditional research we normally do in Nigeria. I will use ABU and BUK because that's the two universities I'm very close to. Like in the pharmacology unit, that's how you must see most of the research, or a lot of them you'll be seeing like a lot are more related to like a drug discovery, and like in pharmacognosy as well, like analyzing a lot of herbs, roots, leaves, and trying to discover anti every day, every year. Anti meaning like anti-malarial,
Dozens of
anti-diabetic.
PhDs on
Yes,
antis.
yes. So, sir, but to my knowledge, sir, those research outcome doesn't get to fruition, or I would say they always stop at Viva. I mean,
That's right.
sir, what's like, why are they not, those things are not well, I mean, taking them further? Like, why do they stop just on the thesis book? Like, why are they
Okay.
not, like, you can see like a very promising research, but it always stop on the thesis, like on the Viva, and that's all. Nothing like that, nothing afterwards, like to
Yeah, I
advance.
understand. Yes.
Yes, sir. So, so you're
Like,
in the
like
field, sir. Any, any
Yes,
big
that's
reason?
it's a very good question, and I think I try to say a lot about this. One lecture I provided for about RTC at the point, I discussed a lot about some of these challenges, and I would say that the problems are generally multifactorial. Like you said, truly, I kept saying we keep doing and producing PhDs, tons of PhDs, dozens of PhDs on anti this, anti this, anti this, anti this,
Yes,
and it stop at that. And that's all. We don't produce anything out of that, like you said, apart
exactly.
from the Bible. Yeah,
Mm,
it's very worrisome. Now, I will tell you that the problem is, in my opinion, multifactorial. Number one, let me pick number one. Number one problem is, let me start with us, then we'll come to policy and other institutional problems and what have you. Let's start with us as researchers, as PhD students. Let's start with us. Now, first we need that exposure to know and appreciate that after this is all about, you live in Australia, everything is about making money.
yes,
Everything is about making money. You
it's true, sir.
understand? About money. So how do I first our exposure? How do I now move this beyond where I found it? So we must change first our psyche. That is, I think, for me, number one. We must change our psyche that this thing is not about uh, about getting degree. It's about solving practical problem. It's about solving problem. Generally, I don't want to go into policy issue. I will talk about it later. Generally, the problem is, let me restrict to our own. We just need to change our psyche to know that knowledge production and knowledge generation is not about getting degree. It's about contributing to knowledge-based economy.
Mm,
A lot of us, I keep saying that even everybody if I'm giving my way, I will introduce a course called knowledge-based economy to every single course, a postgraduate course in Nigerian university, so that everybody should have to see how his work will contribute to economy. So I think, in my opinion, our basic understanding as researchers of that we have a role to play to contribute to economy is now it actually contributes to that problem. That is why you see somebody will produce an very interesting result. You see an exceptional, let's say for instance, an exceptional bowel activity, let's say an exceptional anti-diabetic activity, but it is thrown out. It is thrown out because he is not interested. He never thought that he can now convert that to make money. So that is a mindset issue, and it's a very big one. I think for me that is the first thing. Now the second point, I like I said, I will categorize it into three: us as researchers and scientists, institutional issues, and also policy issues in the country. Now, for us also as research scientists, we also have to do most of the research that we do are not robust enough to be considered for future studies. That is also another perspective. A lot of the research are not robust enough to move to the next stage. That is one key thing. I think one of the presentations, I can remember where I talked about, I will give us an example. If you go to, if you pick a plant, let's say you pick moringa. Moringa is well known. Let's say you take Pachyia biglibosa, that is Urawa. If you take Urawa, you see somebody in pharmacy will study anti-venom potential of it. You see somebody in physiology will study anti-uterine, something that has to do with uterine contraction with Pachyia biglibosa. You see somebody in microbiology will test its antifungal. You understand? Chunk of studies on the same plant, but going nowhere. So I keep saying, if, for instance, within School of Pharmacy, for instance, in pharmacology, they have tested that the Pachyia biglibosa is exceptionally active against a particular, let's say, particular maybe gram-negative bacteria, for instance. Now it's now left for the people in Ceutics to take the same plant and see what they can do within the field, so that nobody will in Ceutic. You see, somebody will also take another plant. Let's say will also take Derivatia and test. Meanwhile, you have one that somebody has done. Then you take it maybe to say you want to check for its ability to break communication of the same fungi. So you see, we are moving forward, same plant, expanding the antimicrobial screening. Then somebody in farm care will now pick it up, and we will now say, you will now isolate the active compound. Then when you put all the results together, then we'll be able to see whether we should go along drug discovery perspective. We'll go along using this formulation, or we should go with a standard with one single compound. Then from there, we will now communicate with Ceutics to discuss about formulation from the same plant. We discuss about formulation from the same plant. Then we move further. Then we can, even if it is something, since we are talking about antibacterial, even the pharmacologists can go and check whether this will have better application topically or orally. You understand? Oh, it has both. These are all things that should be completely looked at as a system, not in silos. But you see somebody looking at formulation with another plant, somebody in farm chem looking for active compound in another plant, somebody in pharmacology testing bioactivity in another plant. You understand? There is no coherence. There is no this. So I think in my and even within the studies, for instance, in the case of pharmacology, for instance, you see the study will not unequivocally demonstrate that bioactivity, even within, or in the case of farm chem, you see they don't characterize the compounds unequivocally. You understand? So I hope you can see there is multifactorial. First, in my opinion, we need to now start sanitizing our effort. Since I'm talking to pharmacists, I think this is the way to go. We look at one, we exhaustively until we produce something from it. If we are working with plants, that is one way to go about it. I talk much, sorry. The other, I can say a lot about examples with this. So you can see based on this, I've given us example with our mind shift. Mind shift also have given us example about as a department or faculty, we do things in silence without properly looking at it. And even within departments, some of the studies we do are not quite robust that to demonstrate particular result unequivocally. That is that. Now, institutionally, sincerely, the institutions also do not give platforms that will allow scientists to make their work proceed better, proceed faster. I mean, the support systems are just not much there. You understand? If you have finding, then you start having complications here and there. First, there is no support. If the support is there, there is complications here and there within the institution. I think that contributes to some of the problem. Because of time, I try to not to expand this. Also, policy issues in the country and also not helping matters. The policy issues. There are no good. Oh, I will say, let me be clear. I will usually say Nigeria has so many good policies on paper, but it's the implementation of policies. So I think as a country, we are not good at implementing so many interesting policies that we have. So policy-wise also, we don't have policies that really, really tie up a lot of research activities into knowledge production and also into knowledge-based economy. We just have to do that so that everybody working for a PhD will have to directly, not indirectly, show that his work is contributing, is producing knowledge, and that knowledge is also contributing to knowledge-based economy. That is a policy issue that is quite missing in the country. And I think if all these things are put together, we may begin to see. So this is the reason why perhaps in NAPRID, they are just doing research, even though it's research and development, but the number of drugs they develop, in my opinion, far outweigh the funding they receive and also the duration of the existence of that institute. You understand? I know they have developed sickle cell drugs and what have you. Yes, very quite innovative, very useful. But what I'm saying, that can be done faster and better if these policies are available or are implemented. Because like I said, we have so many policies but not very much implemented. So I think that is my response generally. Thank
All
you.
right. Thank you so much, sir. I appreciate it. I'm very, very, very well convinced. And I also the only the one decision that you gave, I think those guys back home, please, can you take this message to your respective schools, especially those from the pharmacy schools, like the intra faculty like the collaboration towards like research on this drug discovery. Thank you so much, sir. And I think we can proceed to the next item. Prof. Mustafa.
All right. Thank you so much, Prof. Auwal, for that insightful response to all the questions. Next, we have a presentation of token of appreciation to your humble self. I would like to call on one of us in person of Dr. Abubakar Sadiq Wada to kindly help or support in presenting this token to our speaker. Dr. Abubakar Wada.
Thank you very much, distinguished ladies and gentlemen. On behalf of the PSI group, I would like to present the certificate of appreciation to Professor Mohammed Auwal Ibrahim for your valuable presentation and contribution to this event. Thank you for sharing your knowledge and inspiring us all. Kindly accept this certificate as a token of appreciation from all of us, and we sincerely express our heartful thank you to Professor for his time and the knowledge he shared with us. Thank you very much.
Okay, thank you so much. I really appreciate the kind gesture. It's quite interesting. Thank you so much. And like I said, I thought as a man, even though you said except if you want to if you want to abandon me, but I still remain a mentor also of the PSI. I still prefer to call myself as a mentor of the PSI because I learn and I'm still learning a lot. Thank you so much. This is quite appreciated. Thank you.
Yeah, thank you so much, Prof. Auwal. I believe Dr. Jatta will also help in ensuring that this little token gets to you through email. Yeah, inshallah, as soon as possible. So the final item on the agenda is closing remark, which will be given by my humble self. So just to wrap it up, on behalf of the Pharmacy Scholars Initiative and the Academic Planning Unit, it is my honor and privilege to extend our heartfelt appreciation to our esteemed speaker, Professor Mohammed Ibrahim Auwal, for taking this time to share with us a very great knowledge and experience on AI and drug discovery. Prof. Sir, your enlightened presentation deepen our understanding and inspire all of us to pursue excellence and innovation. We actually look forward to extended collaboration beyond today in terms of research, in terms of mentorship. Now you're talking about you are also a mentee. So we look forward to all working together in that regards. Thank you so much.
Always available. Thank you.
Yeah, and may God bless you. So special thanks to our host, the president of PSI, Dr. Ibrahim Jato, who is the powerhouse of this platform, the Jagaban, I call him. Thank you so much, sir, for bringing us together. Special appreciation to our chairman, who is unavoidably absent till end of this program, Dr. Sha'aban Abubakar, for putting all this together, organizing this, and ensuring that this program is a success. Also, we want to extend our sincere gratitude to all the participants. We apologize for starting very late. We believe that in the future, we'll make sure we make appropriate and clearer communications looking forward. So thank you so much for joining, and looking forward to seeing you again in our future programs. So with this, I thank you all. Have a nice day. Thank you so much.
Thank you, everyone.
Okay. Thank you so much and have a nice day.